Dear students, be welcomed in my first lecture of the year 2021, which will deal with the second part of a digestive system. As you can read here, I will speak mainly about the stomach and about the gut or intestine. Let me start with a short repetition of a general structure of the digestive tube wall. I know that it was a main part or one of parts of the previous lecture. Nevertheless, I would like to repeat it now. Digestive tube is typically composed of 4 layers, as you can see in this schema. The first of them is tunica mucosa, or if you prefer the English terms, mucous membrane. The second layer is a Latin tela submucosa, or you can say just submucosa or submucosal connective tissue. The third layer is called tunica muscularis, or muscular layer. Sometimes there is possible to add the word externa because, as I will say in a moment, the digestive tube contains also inner thin layer of the muscle tissue, and so to be sure it's well to use full name tunica muscularis externa. And the last layer is usually tunica serosa. Today I will speak about stomach and intestine, so there is really mostly tunica serosa, but some parts of digestive tract, for example esophagus as is here written, but also anal channel or some parts of large intestine are covered by tunica adventitia. I will explain what is the difference of these 2 layers. When we will look at several details of those 4 layers, let's start again from the innermost layer, tunica mucosa. Tunica mucosa of digestive tube is very special by the presence of 3 sublayers. It's exceptional. Normally in all other tracts like respiratory, urinary, reproductive, there are just 2 sublayers of tunica mucosa, but in digestive tract is a 3rd additional muscle layer. So in digestive tract the tunica mucosa is composed of epithelium or lamina epithelialis, which can be sometimes stratified, sometimes simple epithelium. It depends on the level of digestive tube. The second layer is called lamina propria mucosae, and it's composed generally of loose connective tissue, which contains some structures like vessels and glands. The 3rd layer of mucosa is called lamina muscularis mucosae, and it is really unique for the digestive tube. You don't find it in another system, and it's composed of smooth muscle tissue. If well developed, there is possible to distinguish their inner circular and outer longitudinal layer of smooth muscle tissue. So that was tunica mucosa or mucous layer. Tela submucosa is simpler. It's just 1 layer of loose connective tissue. If you compare this connective tissue of submucosa and the previously mentioned connective tissue of lamina propria mucosae, you will see that the tissue of tela submucosa is denser. It is not dense connective tissue, it is still classified as loose connective tissue, but you will find thicker collagen fibers, you will find less cells, mainly wandering cells are not so well developed. Yeah, so there are some signs which say us that the tissue is something between pure loose connective tissue and pure dense connective tissue. Nevertheless, if somebody asks you what's the tissue of della submucosa, please say loose connective tissue. And here again, some vessels are present, glands, not everywhere, only in esophagus and duodenum, and also nerve plexus. Nerve plexus, plexus submucosus, is called after its founder, or its the anatomist who first described it, Meissner's plexus. Plexus submucosus meissneri, and it's composed of vegetative nerves and very small vegetative ganglia. The third layer, tunica muscularis externa, is composed usually of smooth muscle tissue, but as you already know from esophagus, there can be also skeletal muscle tissue. Today there will be stomach and intestine, so there will be only smooth muscle tissue in this layer. It is usually the thickest layer of a digestive tube and is subdivided into inner circular and outer longitudinal layer, and between them is a thin layer, thin septum composed of loose connective tissue containing second nerve plexus, myenteric plexus of Auerbach, which is much bigger, much more prominent than the submucosal Meissner's plexus. Tunica serosa and tunica adventitia, or tunica serosa or tunica adventitia, are the outermost layers of a digestive tube. What is their difference? Tunica serosa is covered by simple squamous epithelium, which is called mesothelium. So tunica serosa is equal to mesothelium plus loose connective tissue with high content of adipose cells, adipocytes. Tunica serosa, with its mesothelium, is situated everywhere where digestive tube is surrounded by a abdominal cavity. The same tunica serosa and the same mesothelium is found also on the surface, on the outer surface of the heart, which is also present in the cavity, of course not in peritoneal but in pericardial cavity, and the same situation is on the outer surface of lungs, which are situated in pleural cavity. There are 3 body cavities, and each of them is lined with mesothelium, so the organs which are situated in these cavities are covered by mesothelium. But if the particular part of digestive tract isn't in the abdominal cavity, for example esophagus, which is in mediastinum, or some retroperitoneal parts of intestine like duodenum or colon ascendens, colon descendens, then there isn't this mesothelium on the surface. There is just loose connective tissue which is continuous with surrounding connective tissue of retroperitoneum or mediastinum, and so we call this outermost layer tunica adventitia in these places. So it was short repetition of general structure of digestive tract, and I think that we can go to stomach. This is a small magnification of a wall of the stomach, and 4 labels here are labels of 4 main layers. So A is tunica mucosa with its 3 sublayers. B is tunica tela submucosa or submucosal layer. You can see that it's quite thin in the stomach. C is the thickest layer and it is tunica muscularis externa, and D is tunica serosa covered by a mesothelium which belongs to tunica serosa. The inner surface of the stomach is not smooth. It is folded, and the folds of gastric wall are called rugae gastricae, or one ruga gastrica as is here written. Rugae gastricae are very high and thick, prominent folds which are not stable. They become smaller and almost disappear when stomach is full of food, and they become very high and prominent when stomach is empty. Their core is composed of connective tissue of submucosa here, and they are covered in both sides by tunica mucosa. Tunica mucosa is in both sides. If you take a section of the ruga gastrica, then you will find 2 layers of tunica mucosa and centrally a core composed of tela submucosa. If you look at the surface of stomach in higher magnification, you will see more details than just these than just these big rugae gastricae. You will see also gastric pits and gastric arrhea. Gastric pits are invaginations on the surface. You can see them here in this schema, which is based on the picture from the scanning electron microscope probably. And gastric arrhea are flat fields situated in between individual gastric pits. Maybe better visible will be this situation in real picture from scanning electron microscope. Gastric arrhea are these prominent, smooth parts of the inner surface. Of course, gastric pits are not so well visible. We can see just black holes which represent the outer openings of gastric pits, but when we look at the section of the stomach, we can see it better. So gastric arrhea are these like hillocks, and they are covered by many openings of gastric pits. All these structures, that means gastric pits and gastric arrhea, that means the whole inner surface of the stomach, is lined with simple columnar epithelium. There is everywhere simple columnar epithelium, which you can see here in both these pictures. This right one hasn't very nice quality, but okay. You can see that the same epithelium is on the surface which is exposed to the lumen, that means on the surface of gastric arrhea, and the same epithelium is in gastric pit. Gastric pit is also called in Latin foveola gastrica. The right picture has a very low quality, but it is special staining. It is histochemical staining by PAS method. PAS reaction was used here because this is the proof of high content of carbohydrates in the cytoplasm of the cells. You can see that the cytoplasm is full of carbohydrates, which is a proof of mucus production. Another proof of mucus production is the structure of these cells in normal hematoxin and eosin staining. You can see that their apical cytoplasm is very light, that it contains light, almost colorless granules. On the other hand, basal parts of the cells is basophilic, and nuclei are quite dark and basally situated. So there are all typical signs of mucus producing cells. So that's important thing which we should know. All cells of the surface epithelium in stomach can not only cover the surface and protect it, form the barrier between the content of the stomach lumen and its wall, but they have also secretory functions. There are also glandular cells, and they are secreting mucus. This mucus is very important for the protection of the epithelium, because the stomach is filled with gastric juice, which has very low pH. It is very much acidic, and the mucus, which is alkaline, protects the epithelium against the aggressive function of acidic gastric juice. So, surface epithelium, simple, columnar, and all cells are the same here. All cells can produce mucus. And this is a common feature of the whole inner surface of the stomach. So all parts of the stomach have the same inner epithelium. But there are some differences of individual parts of the stomach. Probably you know from anatomy what is the subdivision of the stomach, which parts can be found here. We can find 4 anatomical portions. Cardia is quite small area near the opening of the esophagus. Then fundus, which is bright bubble-like area above the openings of the esophagus. The biggest part of the stomach is body, so it is the third part, and the last is pylorus. So cardia, fundus, body, pylorus are 4 anatomical portions of the stomach. But in histology we connect fundus and body into one part, which can be usually called body, because the histological structure of these 2 anatomical portions is the same. There aren't any differences of the histology in fundus and body of the stomach. So from the histological point of view, there are 3 parts of the stomach: cardia, body, and pylorus. What are the differences? The first difference is the depth of gastric pits. Gastric pits are the shallowest in cardia. They become deeper in the body of the stomach, and they are the deepest in the pylorus. The lamina propria mucosa of stomach is very rich in glands, and the 3 parts of the stomach, that means cardia, body, and pylorus, differ also in glands. Cardiac glands are usually tubular branched, and they are almost completely mucous because they contain predominantly mucus producing cells and only small proportion of another types. so-called funding glands or glandulae gastricae propriae are found in the body of the stomach, and they are usually simple, tubular, not branched. They contain 5 types of cells. I will speak about them in a moment. And some of them are serous, some of them are mucous, but some of them are very special, and so it is impossible to say whether funding glands are mucous or serous, so we don't use this classification here in these glands. Pylori glands are similar to cardiac glands. They are also almost purely mucous glands, but they are much shorter because gastric pits of the pylorus are much deeper than these of the cardia. But cardiac and pyloric glands are similar. Both are tubular, branched, and almost purely mucus producing. We can distinguish 3 parts in gastric glands. Instead of the duct, there is a pit, and the secretory part of the stomach, of the gland, is composed of neck, body, and base. It is mostly important in the funding glands because the population of cells differs in the neck, in the body, and at the base of the gland. We'll start in cardia. Cardia is a small part of the stomach where esophagus enters, and so there is very sharp transition of the epithelium. Epithelium of the esophagus is stratified squamous non-keratinized, as you already know. Epithelium of stomach is simple columnar, and here in this place you can see very sharp border of these 2 epithelia. There isn't any transitional zone which can be aligned by stratified columnar epithelium. We can find such zones in many places where epithelia change, but not here. In cardia, the transition of the epithelium is very sharp. Esophagus, as you already know, contains 2 types of glands in this aboral location. It contains esophageal cardiac glands, which are in tunica mucosa. One of them is here. And it contains submucosal proper esophageal glands, which are much bigger, which are here. You can see that the proper esophageal glands can go to short distance into the wall of the stomach. Nevertheless, it is its last location. It doesn't go on to the stomach wall. So the proper cardia has got only mucosal cardiac glands, which are, as I said, branched, tubular, and mucus producing. Here is a higher magnification of the tunica mucosa in the cardia. You can see shallow pits and not very prominent branched tubular glands, cardiac glands. Here is smaller magnification, here is a higher magnification of the same gland. If you look at their cells, you probably can recognize that they secrete mucus, similarly as the cells of the superficial epithelium covering their pits. And now we are in the body or fundus of the stomach. Pits are moderately deep and the glands are very special. They are usually simple, straight, tubular, and they contain 5 types of cells. These cells are called mucous neck cells. Unfortunately, it is here Czech written only, but it doesn't matter. The first type is called mucous neck cells, which means that these cells are situated in the neck of the gland and that they secrete mucus. The second cells of gastric glands are chief cells. Chief cells are typically situated in the deepest parts of glands, in the base of glands and also in their bodies, and they are characteristic serous cells. They contain electron-dense serous granules in their apical cytoplasm, which secrete to the gastric lumen enzymes, mainly pepsinogen, which is the proenzyme of the main gastric protease pepsin. Also lipase is here, and some other enzymes in these granules, and their character is typical for the serous cells. The third cell type of gastric glands is called parietal cells. Parietal cells are very special. Parietal cells don't secrete mucus or serous secretion. Their main function is transport of ions, and the result of this ion transport is production of hydrochloric acid. Hydrochloric acid, HCl, is acid which is responsible for the low pH of the gastric juice. The production of this acid is not easy and is not simple. It needs a lot of mitochondria because it consumes a lot of energy. There are two processes. One of them is synthesis of the hydrocarbon acid, and it's splitting into bicarbonate ion and hydrogen cation. These hydrogen cations are actively transported into the lumen of gland, and the second process is active transport of chloride ions from interstitium into the lumen of the gland, and in the lumen, but still in the contact with the apical surface of the cell, the hydrogen and chloride ions are synthesized or joined together, and hydrochloric acid is here synthesized. So the parietal cells are very special, and they are probably the reason why the glands here, the glandulae gastricae propriae or Fundy glands, are not classified as mucous or serous. There are also two minor population of cells in gastric glands. They are not in the picture, but they are written here. They are enteroendocrine cells and undifferentiated or stem cells. I will describe them later when there will be a picture. I didn't say more about the location of parietal cells. They can be situated everywhere. You can find them in the neck, in the body, and in the base of the stomach. In the neck of the gland, they are together with mucous neck cells. In the basal parts of glands, they are together with chief cells. And in the body, there can be a mixture of these three parts, three types of cells. Enteroendocrine cells can be also everywhere. They haven't any predilect place. Undifferentiated cells are found in the neck again. Neck mucous cells or mucous neck cells, they are typical mucous cells which are labeled here MN. So for example, this is a neck mucous cell. I'd like to show the cell with nucleus every time. So here is another neck mucous cell. We can see dark, flat, basally located nucleus, or here, and apical, light, colorless, almost optically empty granules. Typical picture of mucous cells, and as they are situated in the neck of gland, they are called neck mucous cells or mucous neck cells. Both names can be possible. They differ in the structure and also in the quality of their mucus to the mucous cells of the surface epithelium. Both these cells can secrete mucus, but pH is different. Detailed composition of these two mucus is different. So they are not the same cells as the superficial mucous cells. Chief cells are situated near the base or in the base of the gland. As I already said, they are serous cells. They have got serous granules in their apical cytoplasm. They have rough endoplasmic reticulum around the nucleus and under the nucleus, so they are typically serous cells. When found together with parietal cells, they usually close the parietal cells into something like a pocket, which is communicating with the lumen. Here is the lumen of the gland by narrow hole, narrow junction, which can be called intercellular canaliculus. Intercellular canaliculi join the parietal cells and the lumen of glands. So the parietal cells, when we look at them in the light microscopic picture, seem to be situated in the outer layer of the glandular epithelium, but it isn't true. In fact, the epithelium of gland is just simple, but the nuclei of the cells are in various levels. Here is the electron micrograph of a chief cell. So you can recognize it according to very light spherous nucleus, dense spherical non-fusing granules, a lot of rough endoplasmic reticulum situated around and under the nucleus. These are three typical signs of the serous cell you already know from general histology or from salivary glands, for example. You know from the last lecture. If we look at the chief cells in the light microscope, we can see again a light spherical nucleus with prominent nucleolus. We can see basophilic cytoplasm around the nucleus, and we can usually find eosinophilic granules, not very big, eosinophilic granules in the apical cytoplasm. These eosinophilic granules are often lost because they contain enzymes which are quickly activating after death, and so they quickly disappear by autolysis. Nevertheless, when there is a really fresh material, a fresh specimen of a stomach, eosinophilic granules of chief cells can be preserved. Nevertheless, recognition of these cells is mainly based on the basophilia of perinuclear cytoplasm and spherical light nucleus. Parietal cells are a little bigger than the chief cells, and their basic tinction, their overall general tinction is eosinophilic because they contain a lot of mitochondria. As I already said, they contain this mitochondria because they need a lot of energy for splitting of the hydrocarbon acid and for the transport of hydrogen ions and chloride ions. Both are transported by active transport. Here the chloride ions are transported through the whole cell. Hydrogen ions are transported through the apical cell membrane only. Nevertheless, both these transport need energy. To allow the high rate of this transport, the area of the apical membrane is highly increased. It's increased by formation of microvilli, and these microvilli don't only protrude into the lumen of gland, but they are also situated in deep invaginations, which are called intracellular canaliculi. So there are intercellular canaliculi, where the parietal cells communicate with the lumen of the gastric gland. It was in one of previous pictures. And there are also intracellular canaliculi, which are in the cell, which are deep invaginations of the apical plasma membrane, and which are, when you will see them in the electron microscope, for example here, which are full of prominent microvilli. I skipped light microscopic picture of parietal cells, so let's look at it once more. Parietal cells are quite big, usually almost spherical, eosinophilic. You can see there is this prominent eosinophilia. Nuclei are also spherical, but usually a little bit darker than nuclei of chief cells. So big eosinophilic cells, which are found everywhere in the epithelium of glands, of fundic glands. That means they can be between chief cells or and it is this case here, and if they're present in the neck, they can be surrounded by mucous neck cells. Let's look at several electron micrographs of parietal cells. You can see a spherical light nucleus. You can see very many mitochondria, which are the most important organelles of this cell. And you can see quite complicated, because not straightly sectioned, intracellular canal or canaliculus, one and second on the other side, which is full of microvilli, and the same microvilli are also on the apical surface, and all these microvilli are important for the high rate of ion transport, because the higher is the area of the apical plasma membrane, the higher can be the amount of ion pumps. Sometimes you can find a resting parietal cell where you'll find shallower and simpler intracellular canaliculus, which has only several small microvilli, but you can find a lot of so-called tubovesicular structures in the cytoplasm of these cells. These tubovesicular structures is in fact invaginated plasma membrane of the cell, which contains ion channels and ion pumps, and here in this moment it doesn't work, so it isn't situated on the surface, but it is invaginated inside, and it is waiting for new activation of this cell. After activation, these tubovesicular structures change again into microvilli. That means actin microfilaments will participate in this transformation, and microvilli will then fill the intracellular canaliculus of the cell when active. So the parietal cell can undergo some functional cycle. It can be active or resting depending on the needs of hydrochloric acid in the gastric lumen. Parietal cells don't only produce hydrochloric acid, they can also synthesize special peptide, which is called intrinsic factor. This intrinsic factor is secreted into lumen and transported together with other components of gastric juice into the intestine, and it is very important for the absorption of vitamin B12. Vitamin B12 is necessary for hemopoiesis, and so the disorder exists which is called pernicious anemia, where problems with gastric mucosa and the lack of intrinsic factor leads to the problems with hemopoiesis. Mostly erythrocytes are damaged. They are produced bigger, so-called megalocytes, and that's why this called megaloblastic pernicious anemia. You will hear about this problem in clinics, but it is interesting disorder when the disease of stomach can cause the problems with hemopoiesis. So parietal cell is here, and we can see also its electron micrograph again. It is the active form of the parietal cells, where are many microvilli, not only here on the surface but also in the intracellular canaliculus. And the last picture can show inactivated resting parietal cell, where several microvilli are in the lumen of the intracellular canaliculus, but much more plasma membrane is invaginated into these tubovesicular structures, which are waiting as a depot of ion pumps to the new activation. So these were three main types of glandular cells in the body of stomach. Enteroendocrine cells are not specialized or are not special cells of the stomach. They can be found in the epithelium and glands of almost whole GIT. They are found in stomach, in small intestine, in large intestine. They are secreting special peptides or biogenic amines with hormonal activity. And there are many types of enteroendocrine glands. Nevertheless, you can find some general features of them. Their nucleus is usually quite dark because the rate of proteosynthesis here isn't so high. Their cytoplasm is light basophilic. They contain granules, but they are usually invisible in the light microscope because they are too small. But you can see them in the electron microscope. They are situated in the basal part of cells because the secretory surface of these cells is the basal surface, the surface which is covered by basal lamina. You can see it here. Some of these cells don't communicate with the lumen. They are covered by processes of another cells of gland or superficial epithelium. In this case, we call it closed enteroendocrine cells. But another enteroendocrine cells are open. That means they communicate by the apical surface with the lumen. In this case, they have some microvilli, and they can be directly stimulated by content of gut or stomach to secrete their products. There are many types of enteroendocrine cells. They are recognized mainly according to the type of hormone which is here produced and secreted. You can find similar tables in your textbook. I think it isn't necessary to study all the types of enteroendocrine cells. Maybe at the level of the first year histology, it can be sufficient to know where are they found and what is their common feature, or what are their common features. That means small granules, light basophilia of cytoplasm, dark nucleus. Another picture showing distribution of enteroendocrine cells in the GIT. You can see that some of them, like cells secreting somatostatin, so-called D cells, are everywhere. So they are found in fundus, body, pylorus of the stomach, duodenum, jejunum, ileum, colon, so along the whole GIT. But another so-called, for example, so-called GR, enteroendocrine cells are found only in very limited area of the stomach, or there are some enteroendocrine cells which are only in duodenum and jejunum. Yeah, but I think that it isn't so crucial to know their distribution at this moment. As I already said, undifferentiated or stem cells of the stomach are situated in the neck. So here is the area of stem cells. They are dividing, so you can find mitotic figures in them, and they are then migrating and differentiating. If they migrate apically towards the gastric pit and towards the surface of gastric area, they differentiate into the superficial epithelium of the stomach. That means into the mucous cells forming the superficial epithelium. Mucous cells are very, very much attacked by the gastric juice, and so their lifespan is short, about 4 to 7 days. So this renewal here in the surface of the gastric epithelium is very, very rapid. On the other hand, another stem cells after division start to migrate deeper to the mucosa, and they differentiate into all types of cells in the gastric glands. That means if it is fundic glands, they can differentiate into the mucous neck cells, into the chief cells, into the parietal cells, or into the enteroendocrine cells. And in all cases, their differentiation and their life can be longer than the life of the cells on the surface. They can survive several weeks. I will speak about the same undifferentiated cells in the intestinal wall. They have another location here. They are much deeper in glands. So the position of stem cells is one of important differences between stomach and intestine. But let's wait for the same picture in the intestine. So, undifferentiated cells were the last cells found in the gastric glands. Let's quickly say something about the pylorus. As I already said, the gastric pits here are the deepest, which means that the glands are quite short. They can be branched and they are mostly mucous. Only rarely you can find their parietal cells. Very numerous are enteroendocrine cells, mainly these which are secreting gastrin. G cells are here very numerous. On the other hand, you usually don't find any chief cells here. Another important feature of pylorus is thickening of the circular layer of tunica muscularis. I didn't say at the beginning of description of stomach that its tunica muscularis externa has one additional sublayer. In the general description or general building plan of the GIT wall, there were just two sublayers of tunica muscularis externa, inner circular and outer longitudinal. Stomach is an exception. There is additional inner plexiform layer where arrangement of smooth muscle cell bundles is less regular. They form something like a network, and that's why it's called plexiform. That means middle layer is circular and outer layer is longitudinal, and in the case of pylorus, the middle circular layer is very thick and it forms pyloric sphincter. Here is a higher magnification of a pyloric mucosa and part of its muscularis. You can see very deep gastric pits and so only short branched mucous glands. Okay, and we are in the intestine. Intestine is small and large. I hope you know it. Small intestine or both intestines fully fulfill the general building plan of the digestive tract. So they have got 4 main layers which are written here at the right side of the picture. The inner relief of the mucosa is complicated similarly as was the relief of the gastric mucosa, maybe even more than in gastric mucosa. All these complications, all these foldings of the mucosa surface are here because there is necessary to increase the area of the inner surface. Inner surface of the small intestine is important for absorption. It is important for absorption of nutrients, water, and other substances situated in the food. The higher is the area of mucosa, the higher can be the rate of absorption. It was counted that the total area of the small intestinal mucosa is about 600 times bigger or larger than in the case that the intestinal wall is smooth. There are 3 levels of this area increase. The first of them, which is increasing the intestinal surface approximately 3 times, is formation of large folds, which are called plicae semicirculares, or sometimes you can find only circulares, circular folds of Kerckring. These folds are similar to gastric rugae. Their core is composed of tela submucosa, and they are in both sides covered by the full tunica mucosa. They increase the inner surface about 3 times. Then there are smaller finger-like protrusions, which are called intestinal villi or villi intestinales, which are approximately 1 millimeter in length. Their core is composed of lamina propria mucosae, and they are covered with epithelium only, and they can increase the luminal surface approximately 10 times. So there was 3 times, now 10 times, so we have 30 times increased intestinal inner surface. And it's still not all. When I will speak about the cells forming the epithelium on the surface of intestinal villi, you will see the brush border. You already know what is the brush border. Brush border is a very prominent layer of microvilli on the surface of a cell, and this can increase the area of the plasma membrane covering these epithelial cells about 20 times. So if you count everything together, you'll get 600 times higher absorptive surface compared to situation when hypothetically the inner surface of intestine is completely smooth. Here is a small magnification of the small intestine, and you can see circular plicae. It is one circular plica, there is a second circular plica, and they will repeat. Of course, they are prominent in the empty intestine. They are lower in full intestine, but they are quite stable. They are more stable than gastric rugae. The second level of the luminal increase are villi. Villi are these protrusions, these finger-like or sometimes leaf-like protrusions going from the semicircular plicae to the lumen of the intestine. Here is a higher magnification of intestinal villi, so one intestinal vilus. So if you speak about one structure, you say vilus. If you speak about many these structures, you use the term intestinal villi. Intestinal villi are, as I said already, about 1 millimeter in length, about 0.1 millimeter, that means 100 micrometer in width. Their core is composed of loose connective tissue of lamina propria, and their surface is composed of superficial epithelium of the intestine, which is simple columnar. The character of the intestinal epithelium is the same as the character of gastric epithelium. Both these organs have got simple columnar epithelium. Nevertheless, the cells are different. Gastric epithelium has got only mucous cells. On the other hand, intestinal simple columnar epithelium is composed of several types of cells. So it isn't so monocellular. The right pictures are a cross-section of intestinal villi. You can see that sometimes their cross-section can be spherical. In this case, we say that it's like a finger, so finger-like. Sometimes the cross-section of the vilus can be flat, so we say that such vilus is leaf-like. Such flat villi are situated mostly in proximal parts of intestine, in duodenum. Then when we'll go to jejunum or ileum, we'll find more and more finger-like villi. And the last picture here downstairs is from scanning electron microscope, and I think it is the best for the three-dimensional three-dimensional view of intestinal villi. So you can see that there are really processes which are protruding to the lumen. As I already said, surface epithelium of the intestine is simple columnar, and you can see here that there are 5 cell types again. The main of them are enterocytes. Enterocytes were the first cells you met in our department. I hope you remember this. They are typical epithelial columnar cells where you can clearly recognize their basally or centrally located nuclei, their eosinophilic cytoplasm. They have well visible borders with neighboring cells, and they are equipped with brush border. Brush border in this magnification is almost invisible, but for example here you can see something like a line on the inner surface. So brush border is here. Of course, brush border is perfectly visible only in the electron microscope because it's composed of microvilli, which are small structures with the width under the level of light microscopy, and so recognizable only in the electron microscope. I will show them in a moment, and you know them. So enterocytes are the most frequent and predominant cells of the surface epithelium. Their predominancy is the highest in the proximal parts of the intestine, that means in the duodenum, jejunum. Then in ileum you can find more another cells, goblet cells, M cells are in ileum as well, and at the end of the large intestine sometimes goblet cells can be more frequent than enterocyte. But we are still in this in the small intestine where enterocytes prevail. The second cells, which are well visible also in this picture, and you also know them from our practical trainings, are goblet cells. Here is one goblet cell, here is another goblet cell, another one. You recognize them according to mucus production. That means their apical cytoplasm is full of light, big, fusing mucous granules. Their nuclei are situated basally. They are usually darker than nuclei of neighboring enterocytes, and the function of them is to produce mucus. The third type are M cells. They are special antigen presenting cells, and they are found only in these places where lymphoid nodules are under the epithelium. You will find them mostly in the ileum. I will show then some picture from ileum where M cells are situated. Then enteroendocrine cells can be found here. They don't differ from these cells in the stomach, which were already described. And the fifth cell type here are tuft, or also we can say brush cells. They are quite new. They were recognized only several years ago, and they serve as chemoreceptor cells. And it isn't easy to recognize them at the level of light microscopy, and that's why I can show them only in one electron micrograph. So let's start from the most frequent enterocytes. Enterocytes are also called absorptive cells because their main function is absorption of nutrients, water and other substances. Their cytoplasm and their outer structure can differ according to type of nutrients and location, so they can be different in case that they just absorb mostly water or mostly lipids. Nevertheless, the most prominent organelles here are usually mitochondria. That's why they are eosinophilic. Their cytoplasm is eosinophilic, but they contain quite well-developed Golgi complex as well. They contain well-developed rough endoplasmic reticulum because they must produce proteins. Some of the proteins which are here produced are transported into the apical plasma membrane, and they become a part of this apical plasma membrane. They are usually glycoproteins, that's why they are transported through the Golgi complex, and they occupy the area of brush border, and they form additional layer on the surface of brush border, which is called glycocalyx. This glycocalyx is a layer of glycoproteins which are firmly connected with the cell because they are integral membrane proteins, and they have enzymatic function. Some glycoproteins of the glycocalyx can be disaccharidases, and other glycoproteins here can be peptidases. So enzymes which are important for the digestion of sugars and proteins. So proteins and sugars are not digested only by secretory enzymes like amylase or pepsin from stomach or trypsin from pancreas, but also by peptidases and saccharidases in the glycocalyx directly on the surface of enterocytes. Here is the small magnification of electron micrograph. Nevertheless, even in this small magnification, we can see individual microvilli of the brush border. We can see mitochondria in the cytoplasm. We can see Golgi complex typically located above the nucleus. Nucleus is here. And again, mitochondria under the nucleus. This area can contain also rough endoplasmic reticulum. There is a detail of brush border in longitudinal and cross section. You hopefully remember that microfilaments are necessary for the supportion of brush border of microvilli here, and that they are stabilized by several actin binding proteins to allow the supporting function. And here is a picture showing glycocalyx, which is in the area of a brush border and above the brush border, and which hasn't only protective function but also enzymatic function. For example, sucrase is one of enzymes here. It digests sucrose or isomaltase. Also lactase is here, which digests another disaccharides. Goblet cells are mucous cells, and I think it isn't necessary to speak more about the structure of mucous cells. You know them from general histology and from salivary glands, and you will see them again in the respiratory tract and in another tract. So mucous cells are well known. They have apical light cytoplasm containing granules and basal quite dark nucleus. They secrete mucus and their number is increasing in distal direction. The third cell type, which is specific for the intestinal epithelium, are M cells. They are called M because they have got so-called microfolds on the surface. Instead of brush borders, there are microfolds, something like a three-dimensional network composed of low folds of plasma membrane. They are antigen-presenting cells. They can recognize and bind antigens which are coming to the intestine together with food. They can digest these antigens, which leads to their inactivation. So if that's something dangerous, it's not more so much dangerous after this digestion. And then they present individual epitopes of these antigens on the surface of their basal membrane in the complex which MHC2 complex. MHC is important immunological complex which is particularly this MHC2 is typical for the antigen presenting cells. I think that you know it from lymphoid system, which was in one of previous lectures. The presence of antigens in the complex with MHC is a signal for lymphocytes to penetrate through the perforated basal lamina here. They penetrate to the epithelium. They become situated in deep pockets, in deep invaginations of the basal plasma membrane of M cells. They come to the contact with the surface of basal plasma membrane and they acquire information about the antigens situated in the intestinal lumen. After that, they can leave back, they can penetrate through the perforated basal lamina back to lamina propria, and if stimulated, they can activate another cells. These lymphocytes are usually T helper lymphocytes. You know that T helper lymphocytes are of several types, and they can activate macrophages or B lymphocytes or granulocytes. You know that there are several types of helper T cells. So microfold cells are important for the communication between the lumen and the content of the lumen of the intestine and our lymphatic system. So they are present usually at these places where lymphoid nodules are present in the intestinal mucosa, and the typical place where are many lymphoid nodules is ileum. So the most microfold cells are situated in the ileum. The recognition isn't easy. The author of this picture labeled two cells that they are M cells, but in fact I don't know how did he recognize it. I can recognize the M cell only in the case that it has just some lymphocytes in the pocket on the basal size, but here nothing like this is situated. I see many lymphocytes under the epithelium. All these dark cells here are lymphocytes, so it is a part of lymphoid nodule, and these lymphocytes migrate to the epithelium. You can see it here or here or here. Here are lymphocytes migrating to the epithelium, and some of them are migrating into the contact with the basal membrane of M cells. So in this case, as it's visible here in this schema, the M cell should be recognizable. If you see a cell with its nucleus and also several small lymphocytes in their very close vicinity. But not all lymphocytes in the epithelium mean that they are situated in the invagination of basal plasma membrane of M cell. Lymphocytes can penetrate between another epithelial cells, for example between enterocytes, so the presence of lymphocyte in the epithelium isn't proof of the M cell at this place. Enteroendocrine cells of the intestinal epithelium are the same as were these in the stomach. They can be again open or closed. That means sometimes they communicate with the lumen, sometimes not. This electron micrograph, this left electron micrograph, is very, very good because it contains three enteroendocrine cells, 1, 2, 3. We can see basally located granules in all of them, and you can recognize that these granules are different. Here in this cell 1, granules are quite big and moderately electron dense. In this middle cell, granules are also big but more electron dense. And in this right cell number 3, granules are very small. What does it mean? It means that these are 3 enteroendocrine cells, but each of them is of different type. Each of them can secrete some another peptide or amine, some another hormone. I am not able to recognize which hormone is here secreted. You need immunohistochemistry for this detailed study, but the proof that there can be 3 enteroendocrine cells of 3 types in one place, it is. Probably all these 3 cells are of the closed type because they don't reach the lumen. On the other hand, this right enteroendocrine cells is in the contact with lumen, and there are small microvilli on the apical surface. Last cell from the intestinal epithelium is tuft or brush cell, which has microvilli, but these microvilli are a little thicker than you can find on the surface of enterocytes. They have very deep roots composed of microfilaments, and it was discovered that the apical plasma membrane on the surface of these microvilli contains special taste receptors which are very similar or the same as real taste receptors in taste buds on the tongue, and these taste receptors here in the intestine are used for the recognition of bacterial products, and so the information given by the stimulation of tuft cells can be recognized as the signal about bacterial invasion or bacterial infection, and can be sent to the central nervous system, which can be informed about this new situation and can prepare some response, for example, stimulation of immune system. So Taft cells are important for chemoreception, and mostly for chemoreception of bacterial products. Under the epithelium is a layer of loose connective tissue which is called lamina propria mucosae. Not only cells of connective tissue are here, you can find there also smooth muscle cells which migrate here from the lamina muscularis mucosae. That's because the intestinal villi are slightly movable processes. They can move, they can slightly contract, and these movements can mix the intestinal content to allow new molecules come to the surface of the absorptive epithelium. The other cells of lamina propria mucosae are fibroblasts, macrophages, mast cells are here. And there are also many lymphocytes, even in these places where are not lymphoid nodules. All lamina propria mucosae of the intestine is rich in lymphocytes, forming together so-called gut-associated lymphoid tissue. Lamina propria mucosae doesn't contain only connective tissue, but there are also intestinal glands. Intestinal glands are called Lieberkühn's crypts, or crypts of Lieberkühn. Lieberkühn crypts are simple tubular striate glands, usually quite short in the small intestine, but they will be longer in the large intestine. And there are again 5 cell types in these glands. So it's quite simple for the memory. There are 5 cell types in gastric glands. There are 5 cell types in superficial epithelium of the small intestine. There are 5 cell types in the epithelium of Lieberkühn's crypts. Some of these cells are the same as the cells of superficial epithelium. It isn't anything surprising. If you look at the previous picture, you can see that the epithelium of the surface, that means epithelium of the intestinal volus, is continuous with the epithelium of gland. So here is the gland or crypt of Lieberkühn, here is the volus, and epithelium is just one here, but different. The epithelium of the volus contains some same cells, but some different cells than the epithelium of crypt. Which cells are the same? Enterocytes are in both locations, goblet cells are in both locations, and enteroendocrine cells are in both locations here in the small intestine. 2 of these 5 cell types are specially situated only in crypts. They are Paneth cells and undifferentiated cells. Paneth cells are situated in the really very basal portion, in the deepest portion of crypts. They line the bottom of crypts, and you already know them because they were demonstrated as the typical example of serous cells. They have very big eosinophilic granules and basophilic cytoplasm around the light spherical nucleus. They secrete so their granules contain special bacteriocytic enzymes like lysozyme, lactoferrin, defensins. These enzymes are necessary for the maintaining of the bacterial population in the small intestine in a relatively low level. Paneth cells almost don't exist in the large intestine, so the bacterial population there in the large intestine is very high. You maybe know that there can be 1 or 2 kilograms of bacteria in the content of large intestine. On the other hand, small intestine is almost sterile because there are Paneth cells in the crypts which secrete lysozyme and other enzymes which are reducing the bacterial population here. Undifferentiated cells are present also in the base, but not so deeply. They are situated somewhere here in this level near the base of the glands or crypts. Paneth cells, as I already said, are typical by very big eosinophilic granules. I don't know any other cell with such big and such clearly strongly eosinophilic granules as the Paneth cells. These granules contain these bacteriocytic enzymes. These granules or these enzymes are proteins, so the cells need a lot of ribosomes in a rough endoplasmic reticulum, and that's why the cytoplasm here around the nucleus and under the nucleus is strongly basophilic. Nucleus is light because the level of transcription is here very high. Instead of the Paneth cells, you can see here several enterocytes with brush border. You can see one goblet cell in this section of crypt of Lieberkühn. So you can see that these glands contain really several cell types, and similarly as the gastric glands, we don't use here in the intestine the classification serous mucous, seromucous glands, because not only serous and mucous cells are here, but also enterocytes and enteroendocrine cells, which are neither mucous nor serous. So we don't use this classification here. We can use just morphological classification. Intestinal glands are simple, tubular, striped. Electron micrograph of Paneth cells is showing electron-dense big granules, so the same granules which are eosinophilic in the light microscope are electron-dense in the electron microscope. There is a spherical, basally located light nucleus, and higher magnification can show a rough endoplasmic reticulum mostly around this nucleus. As I already said, undifferentiated cells in the intestine are in the crypts. They differentiate into all types of the intestine. That means into Paneth cells. The life of them is quite long, about 1 month, and they can differentiate also in the goblet cells or absorptive cells or enterocyte, and the length of life of these cells is lower, less than 1 week, similarly as the lifespan of the superficial cells of the stomach. Cells are migrating towards the tips of villi, and then they are desquamating and they are renewing by cell division and differentiation of the undifferentiated stem cells in the deepest parts of the crypts. The other things which you can find in lamina propria mucosae are vessels, blood vessels and lymphatic vessels. Then there is lamina muscularis mucosae, which is composed of smooth muscle tissue. Then stella submucosa, it is composed of loose connective tissue, little denser than this in the lamina propria. Again, vessels are here, and also nerve plexus of Meissner. Vascular bed of the small intestine is in this picture. There are several plexuses. Capillary plexus is in the core of each vilus. Second plexus, which is composed of venules and arterioles, is in the lamina, in the tela submucosa, and the plexus of biggest veins and arteries is in the serosa layer. Lymphatic capillaries are blind structures situated in villi again. They can be sometimes dilated because mostly lipids are absorbed directly to lymph as so-called chylomicra, and in this case the lymph after absorption of lipids can be like milk, white, and that's why these lymphatic capillaries of intestinal villi are called lacteals. Exceptional additional structure of duodenal submucosa are Brunner's glands. Brunner's glands are branched tubular mucous glands which are specific for the tela submucosa of duodenum. They secrete mucus with high pH, which is important for the neutralization of the intestinal content which is coming to duodenum from stomach, and it is acidic, and so it has to be neutralized by alkaline mucus from duodenal Brunner's gland. Brunner's glands are very prominent. They can perforate the muscularis mucosae at this level and penetrate to the lamina propria. They haven't their own ducts. They communicate with crypts of Lieberkühn here and empty to the surface together with crypts. The specific feature of mucosa and submucosa of ileum is are many lymphatic nodules. They form aggregates where sometimes up to 200 nodules can be found in one place, and these aggregates of lymphatic nodules of the ileum are called Peyer's patches. Peyer's patches are typical for the ileum. The mucosa above Peyer's patches doesn't contain many villi, or if yes, these villi are only low, small, short, and the mucosa surface is mostly smooth, and here this is the place where are found M cells. So M cells, microfold cells, are found mostly in these smooth mucosal areas above the Peyer's patches. You can see that some circular fold with normal villi can be found in the ileum as well. These Peyer's patches are found only in the part of wall of the ileum, mostly at the side which is opposite to the mesentery. Here is a detail of mucosa in the ileum with Peyer's patches. So Peyer's patches are aggregates of lymphatic nodules, and mucosal surface above them is smooth or forms something like such domes here. And here in these locations I would look for M cells. Tunica muscularis externa of the intestine has typical double composition, inner circular and outer longitudinal layer. Their thickness is almost equal in the small intestine, and between them there is a thin layer of loose connective tissue with many nerves and nerve ganglia, which together represent myenteric Auerbach's nerve plexus. This nerve plexus is important for the innervation of smooth muscle here in this location. So it means for the innervation of tunica muscularis externa. On the other hand, smaller submucosal nerve plexus, which is located in this location here. You can see that this is a duodenum, so there is also Brunner's gland, but at the same location can be elements of submucosal plexus. So it is important for innervation of muscle tissue in the lamina muscularis mucosae, and also for these smooth muscle cells which are dispersed in the villi of the lamina, in the lamina propria of the villi. And as the muscular layer of muscularis externa is much thicker, logically myenteric plexus is more developed. Nevertheless, in both cases, this is the network of sympathetic and parasympathetic nerves, and together with them there are ganglia. Ganglia, when situated in the wall of digestive or another hollow organs, are mostly parasympathetic. Sympathetic ganglia are not situated so peripherally. They are found around the vertebral column in the sympathetic chains, truncus sympathicus, but parasympathetic ganglia can be located very peripherally as so-called intramural ganglia. So you can find them in Meissner's or myenteric plexus as well. All these ganglia and nerves here are innervating muscle tissue, and also glands, Brunner's glands or crypts of Lieberkühn are innervated by sympathetic muscles, sympathetic and parasympathetic nerves. Here is a detail of Auerbach's plexus. Not very large quality of the picture, but I can recognize vesicular nucleus of a neuron here or here. So it is a ganglion, parasympathetic ganglion in the myenteric plexus. Myenteric and submucosal plexus are not alone in the responsibility for the movements of the intestine. Regular, slow peristaltic movements can be generated by so-called interstitial cells of Cachal, which are situated directly in the wall of the intestine. You can find them in the smooth muscle tissue or in the connective tissue of the submucosa. There is still discussion whether these cells are more transformed neurons or transformed smooth muscle cells, but currently the second possibility is preferred. So probably they are transformed or specialized smooth muscle cells which are connected by processes together and also with real working smooth muscle cells, and they generate slow waves, slow slow action potential, similarly as the cells of cardiac conducting system can generate fast action potentials necessary for the autonomic heartbeat. So there can be some similarity of special cardiomyocyte in the heart conducting system and these interstitial Cajal cells of the intestine. So the peristaltic movements are generated by both nervous system and autonomic nerves and ganglia, and by interstitial cells of Cajal. And now we are leaving the small intestine and we are entering large intestine, which is shorter and which has many similar things to the small intestine. So I will speak mostly about differences here, and I will not describe all the structures here once more. General structure is the same, 4 layers. Of course, some parts of large intestine are secondary retroperitoneal, and so their outer layer can be tunica adventitia. It is the case of colon ascendens and colon descendens, where only ventral side is covered by serosa. The main difference of small and large intestine is the inner relief of the tunica mucosa. Large intestine doesn't contain any villi, and so its inner surface is in fact smooth. We can see some folds. We can say still semicircular or circular folds are here, but not villi. No villi are in the large intestine, and so the crypts of Lieberkühn are much deeper than these which were visible in the small intestine. So, main differences of large and small intestine: no villi in the large intestine, deep crypts of Lieberkühn, and also the longitudinal muscle layer is different. You can see that there is the thickening of longitudinal muscle layer here, and if we see the whole lumen of the large intestine, we would see 3 thickenings. They are called teniae coli. You know them from anatomy. So teniae coli are bundles of longitudinally arranged smooth muscles, but remaining parts of the intestinal wall contain only very thin layer or reduced, almost no layer of longitudinally arranged muscle. Of course, circular muscle is developed, and it is of the same thickness in the area of the tenia and in the area between teniae coli. Submucosa doesn't contain any glands, but it isn't anything surprising. So, to say it once more, the main differences of large and small intestine is the absence of villi here, deep crypts of Lieberkühn, and the presence of teniae coli composed of longitudinally arranged smooth muscle. If we look at the details, we can find another differences. For example, enterocytes, sorry for the quality of this picture, enterocytes of large intestine are different. Their brush border is lower, and the number of microvilli there isn't so high. They are specialized for the water absorption, not so much for the nutrients absorption, because almost all nutrients were absorbed by the enterocytes of small intestine before. and so their intercellular spaces are more dilated because water is absorbed through the brush border and leaves the cell not only basally but also laterally. And these enterocytes of the large intestine can sometimes be called colonocytes. So colonocyte isn't any new cell type, it is only special enterocyte. Here are microvilli of the colonocytes, and you can see that they are quite short and that there are bigger gaps between them. The inner surface of the large intestine in the scanning electron microscope can show openings of crypts. This is the opening of crypt. Here is another one, another one, another one. But in between them aren't any villi. The surface is smooth. There are some droplets of mucus which are secreted by goblet cells, but no villi. Section of the mucosa of the large intestine can show also almost smooth surface, no villi, very deep crypts, and much more goblet cells in the epithelium, in crypts and on the surface, in both these locations. The more distal part of the large intestine it is, the more goblet cells are there, so in the rectum goblet cells prevail. Paneth cells are rare here. It is a problem to find them. If you find any Paneth cells, it's usually just single cell in the deepest parts of crypt. And this is the reason why bacteria can survive very well in the lumen of the large intestine, and why there are so many here. Lymphoid nodules can be found frequently in the mucosa or submucosa of large intestine. In this case, this nodule is at the border of these both layers, and muscularis mucosae is here interrupted. But they are never so aggregated as in the ileum. So it is impossible to say that they are also Peyer's patches. No, Peyer's patches are only in the ileum. Here are single lymphoid nodules in the large intestine. The exceptional amount of lymphoid nodules is in the appendix. Vermiform appendix is a derivative of large intestine, but its lumen and its size is much smaller, and their wall is full of lymphoid nodules. Nevertheless, there is still typical structure of digestive tube. You can find all four layers: tunica mucosa, submucosa, tunica muscularis externa, and tunica serosa. Tunica mucosa and submucosa are not fully separated, but lamina muscularis mucosae, it is developed only in several places, but it is mostly interrupted by big lymphoid nodules which are occupying both submucosa and mucosa, and so there isn't any place for muscularis mucosae at these locations. Nevertheless, if you look at the detail of the wall in the appendix, you can find normal crypts of Lieberkühn with some goblet cells, but not so many because we are at the beginning of the large intestine, and everywhere there are many lymphocytes. These lymphocytes are also among the crypts. They are not only in the nodules. Nodules are formed by B lymphocytes mostly. On the other hand, here this diffuse population of lymphocytes, there are mostly T lymphocytes. Taenia coli go towards the appendix vermiformis and join together, so the longitudinal layer of the smooth muscle here is again continuous. There aren't any Taenia coli. There is normal structure of circular and longitudinal muscle, but muscle tissue of appendix is quite thin. In humans, the appendix serves mostly as a lymphoid organ. It hasn't any importance for digestion. On the other hand, in some animals, there is special population of bacteria in the appendix, and these bacteria are important for digestions of cellulose, and so it is very important for these animals which are eating grass or leaves mainly. But human don't belong to this group of animals, and so our appendix is mostly lymphoid organ. It serves as a abdominal tonsil. In fact, it's also sometimes called abdominal tonsil. Okay, the last segment of the intestine is anal channel. It is the area where intestinal simple columnar epithelium transits into the stratified squamous epithelium of anus. This transition here isn't so sharp as the transition between esophagus and stomach, so there can be found a zone, transitional zone, composed of stratified columnar epithelium, but this zone is very, very short. Crypts of Lieberkühn, which are found in this area of anal channel where is still simple epithelium, are very rich in goblet cells, so they almost resemble mucous glands. Then is the transitional zone and stratified squamous epithelium, which goes to the surface, so gradually changes from non-keratinized to keratinized epithelium. Submucosa of the anal channel is very rich in veins. There is very special venous plexus which is called hemorrhoidal plexus. You probably know what hemorrhoids are. It is a very painful disorder when this venous plexus is filled with blood. There is thrombosis and inflammation. That's why this plexus is called hemorrhoidal plexus. It is typical for the submucosa of anal channel. External muscle, that means tunica muscularis externa, is composed normally from smooth muscle cells, but the circular component of this smooth muscle cell is very prominent and represents the inner sphincter of anus. There exists also outer external sphincter composed of skeletal muscle tissue, which doesn't belong directly to the wall of the anal channel. In fact, it is separated from the anal channel by its adventitia. Nevertheless, you can find external sphincter on the slide as well, so you will see there not only smooth muscle but also skeletal muscle tissue. And now we are at the end of the intestinal tube and of the whole digestive tract, so I think that can be the end of my lecture. So thank you for attention and goodbye.